Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
And I'm going to go straight to our Q&A. And I'm going to stick with you, Paul. Although, Deborah, we invite you back into the conversation as well. And we do have questions lined up for you. So stand by. But starting with you, Paula, just to pick up on that very last point you made, because people are naturally interested in whether this can be applied to COVID-19. And one of our alumni and friends asked whether this layering approach could work to repair a COVID-19 infected cell. And it sounded like you were saying you were hopeful in that regard. Tell us more.
We're very interested, in fact, in whether or not we could use this as a way to address that huge cytokine storm that occurs with COVID 19 patients. A lot of the damage and death occurs because of the response to the viral infection as opposed to the virus itself. So, the idea is that if we can incorporate some of the treatments for inflammation, we might be able to lower that cytokine storm more effectively if we target the lung specifically. And it turns out that lung cells, when they're inflamed, also exhibit markers that would allow us to target them more specifically and deliver those drugs directly to the lung. We're also interested in some of the new therapies that are being talked about now and whether or not we could encapsulate those therapies more effectively. And in terms of cell repair, I think that certainly there may be ways in which we could incorporate DNA repair agents that could help with some of the damage.
Thank you, Paula. I'm going to stick with you for a minute because the questions are just rolling in right now. So everyone's very interested in your presentation, and then we'll switch back to Deborah in just a few moments. So tell us about a little bit about how close you are, you and your team, to thinking about commercial scaling of what you're exploring with these nanoparticles. In particular, an alumni asks, what would commercial scale manufacturing and a supply chain look like for these kind of complex nanoparticles?
Yes, actually, we thought of quite a bit about this. We're using the cores that are already commercially available and approved. So liposomes and polylactic glycolic acid nanoparticles. These are two nanoparticles that are already approved for other applications in medicine. And we're wrapping polypeptides, which are generally regarded as safe, around the nanoparticle and introducing our siRNA. So we think that some of the hurdles would include FDA, and we think that we are designing these so that we can prevent a great deal of difficulty in getting FDA approval for something like a clinical trial. In terms of manufacture, I have great students. They're chemical engineers, and as you know, chemical engineers get heavily involved in process. So we have been looking at different ways of processing the nanoparticles so that we can actually have a much more rapid and large scale manufacturing process. And we've been using tangential flow filtration, which is a commonly used pharmaceutical approach for purification as a way of isolating our nanoparticles after applying each layer. And that's actually sped up the process significantly and also allowed us to increase the scaling. So I think we would really work hand in hand with companies that already have a great deal of pharmaceutical processing expertise. And because we tend to work with formulations that are either two layers, which is the case of the ovarian cancer example, or four layers, which was the lung cancer example, that we can actually get this done very effectively and at relatively low cost.
Thank you, Paula. One more question for you. And this is something else you just touched upon when you talked about the engineering, the chemical engineering students looking at the broad array of not just the hard science, but how you would scale it up. Um, we're being asked, what is the range of your team? What kind of disciplines are represented in your team, and what roles do they play in the work you're doing?
Oh, yes, I love that question. One of the things I'm especially happy about, what I love about being at MIT is that I have a broad set of disciplines in my lab. I'm in chemical engineering. My lab is located in the Koch Institute of Integrative Cancer Research, where we share space with biologists and clinicians. In my lab, I have students who are studying biological engineering and biomedical engineering through the IMES HST program as well as chemical engineers. We have material scientists, chemists, biologists. We also have 2 clinicians in our lab who are doing unique postdoctoral studies in which they are working from Boston Children's Hospital and Brigham and Women's Hospital for glioblastoma and ovarian cancer, respectively. So we actually have these clinical experts in our group meetings who are telling us what's useful and what's not useful.
Fantastic. Really great to hear. And thank you so much. Paula, there may be more questions coming in for you, but I'd like to turn back to Deborah if we could. So Deborah, we have a whole bunch of questions that are being upvoted as people have been thinking about your earlier presentation. And what has risen quite to the surface is really a question around how you would compare the confidence levels we have in the safety of domestic food versus food that's imported, a lot more of which we're seeing in our grocery stores these days.
Yeah, that's a great question. I think it depends on the country that's imported from. For instance, I'm skipping from food over to cosmetics now. In general, you'll see people suggesting that they look at getting their cosmetics from Europe because there's a number of compounds that are allowed in US cosmetics that are banned in those produced in Europe. And in the same way, I'm just going to pick one more cosmetic example. I used to write a column for the New York Times about toxicology called Poison Pen. And one of them looked at lead in lipstick. And it was very clear that the lipsticks that contained high levels of lead, you can actually find a whole page about this on the FDA website, were almost routinely from China. So now we're talking about where should we focus our imported inspections? And I think they need to look at the countries from which the imports come. And I think we also need to look at the levels of inspections. FDA is responsible for almost all of those inspections. And as I indicated in that slide, they physically inspect very few of them, but they have come up with a computerized hazard prediction program that I think does effectively sometimes enable them to see what looks like a likely risk. So that's a sort of rambly example saying there are a lot of unknowns in this system, and I think we're working in that regard semi-well.
And so let me just follow up on that and sharpen the question a bit. Is there a resource you would point people to if they want to look at particular countries of origin or think about what they're picking up in the market? How do you feel about, fruit from Chile, for example?
Right. So the real question with some of these imported countries is the pesticides that are allowed in those countries versus the ones that are banned here. And so the environmental working group does a pretty good look at some of the kind of applications of pesticides on imported fruits. The FDA has a imported pesticide section, and you can certainly find some of this. I wish I had assembled all these links in advance, but you'll also find some of this at the EPA, since the EPA is primarily looking at pesticides in fruits. But I would, for instance, if I was concerned about that, I would take a look at some of the FDA restrictions on pesticides that are permitted on imported produce and decide which ones I actually cared about. I might tell you, though, especially in the last few years, some pesticides that are banned in countries like Brazil are now permitted in the United States. So just being paranoid about imported produce doesn't always serve you well. We aren't leading the world in regulating pesticides in every case. So the other part of that is, in this kind of long answer, is that I would also look at produce that is known to have high levels of pesticide in general. Strawberries being a case study, apples being a case study, potatoes being a case study. And because I spend a lot of time looking at toxic substances in food, I don't go running to organic, low pesticide exposure for all kinds of fruits and vegetables. But for those three things, for strawberries, for apples, and for potatoes, I buy organic, right? Just because I know those tend to be in the traditional sense more heavy pesticide products.
Thank you very much, Deb. So, just picking up on that theme on regulation in the United States, a number of people are curious about whether or not the current administration has really cut back resources on food inspection and attention to safety in the food supply. Is that happening, or how are we to think about that?
Yes, that's a great question. So, one of the things that's happened, going back to that slide I put up showing this sort of wonderfully supportive environment for USDA inspections of meat, is last year the Trump administration actually rolled back meat inspections of pork processing plants to pre Upton Sinclair times and removed inspectors, USDA inspectors from a majority of the plant and tipped that over to pork processors to inspect their own meat production lines. And they also allowed pork processors to increase the number of carcasses that could run through a plant. And once they did that, this hasn't happened yet. The beef industry came in and said, well, if you're going to let pork processors to inspect themselves as they used to in 1905, why can't we do that too? And so my understanding is that is in the works. There's a few other things that have been rolled back, and one of the most important ones is that in 2011, the Obama administration passed the Food Safety Modernization Act, essentially updating us, saying, let's use really smart science. Essentially, let's use the ability to work at the nanoparticle part per billion level that you see in the really smart labs at MIT, evidence in a different way by Paula's work. And let's put that into understanding the food supply and what's dangerous. And that particular act, Food Inspection Modernization Act, has also been partially moving forward, but significant parts of that have been put on hold by the Trump administration. I have other examples, but those are two that stand out for me.
So I see Paula nodding there when you spoke about the applications of her kind of work into this kind of work. Paula, go ahead. Tell us why you were nodding so vigorously in that particular moment.
I think there's some opportunities here in what Deborah brought out. And one of them is to actually use nanotechnology for detection of a range of different contaminants in food. Because right now we're seeing that nanotechnology has definitely been impactful in helping us to detect disease. I think we can apply the same concepts, but on a much cheaper level, and create some testing that can be done reliably and allow essentially technology to go hand in hand with policy, as Deborah was describing.
Yes, and if I can just jump in there, I'm so glad you said that, Paula, because it actually is one of the things I've thought about in the way that I do this policy related work. Is that MIT is a wonderful place for this. Not only having the smart science, but being here, I talk to so many people who get that science doesn't exist in some, uh, bubble of a vacuum, that it actually applies to decisions we make about how we live our lives and how we take care of each other. And so it's really exciting for me to hear you say that and to mention what a supportive environment that is for me in the way I think about these issues and try to get this particular message out.
Absolutely. I think it's a great opportunity for partnerships across the fields.
Agreed.
This is the place to do it.
Yes.
This is just so wonderful to hear. Thank you both, because I was going to ask this question, but you went ahead and answered it. And I'm sure that everyone who's participating in this conversation is really delighted to see both of you thinking about how your work relates and connects. But are there concrete examples in the work that's happening, Deb, where the research strength of MIT is really going into the field that you are studying from the journalism side?
Yes, I mean, going back again to being able to measure contaminants in food at the nanoparticle level, or even not, you don't have to get as tiny as that, at the part per billion level. I think, and then this is something that I think has been really pioneered at laboratories at MIT, opens up this whole landscape of ways for us to think about making the food supply safer. And that's because I think a lot of what we're talking about now is not dairymen pouring vast quantities of formaldehyde into food, but chemicals that are at a level in the part per billion or less level, that we're trying to figure out how they function at that level. And we're trying to figure out how to make people safer with what is basically the field of low dose toxicology. And so I have talked to people at MIT who really are sort of unpeeling the layers of how we figure that out. I think it's just really exciting. And you know, at a time that I think we all are looking for positive notes in what's going forward, this is actually a positive note.
Paula, you're nodding and smiling again. I don't know if everyone can see you, but I can. So open the floor to you if you want to add in.
No, absolutely. I think that there's a huge amount of opportunity here, including the chance to look at not only food but water, our water supply. There are a number of technologies that have been developed recently which allow rapid detection of certain proteins, certain metals, and other kinds of components. And they use the ability to couple sort of the binding event with some sort of translation to a signal. And if we're able to maximize that and combine that with what we can do with artificial intelligence to recognize patterns of dangerous foods, for example, or dangerous water, there could be a lot that could be done for environment as well as for food. And on Deborah's point, we have huge policy brains at MIT that can help guide where this technology should go. And I think also help guide our government with these ideas and positions.
I think that's right. And just to pick up on Paul, this is so fun to pick up on Paula's point. You know, people tend to, if they don't really know this territory, to say, well, what does a contaminant at a part per billion really mean? Is that an issue or not? But if you look at, and I know this makes me sound a little creepy, but arsenic is one of my favorite poisons. If you look at arsenic, the EPA standard for arsenic in drinking water, which you were just talking about, water, Paula, is 10 parts per billion. And we know from looking at arsenic contamination of drinking water in other countries, particularly in Asia, Bangladesh, in Taiwan, that if you get much above that, you start seeing actual health effects. In Taiwan, they actually had an incidence of what they called Blackfoot disease because arsenic is damaging to the cardiovascular system. Once you hit about 50 part per billion, the people developed gangrene because they were drinking water that had 50 parts per billion arsenic, and it was so destructive that they started seeing gangrene in their feet. So, to just say, oh, well, it's a tiny amount, doesn't make sense. This kind of science where you can look at things at these minuscule, amazingly small levels and understand that they do matter, I think is one of our futures. I'm excited to see it here.
Thank you both so very much. I'm afraid we're going to have to wrap up here, but just before we do, I'm going to give you a lightning round question, Deborah. Let's call it a bonus round, but you have to give me a yes or no if you can.
Okay.
It's related to arsenic because several people voted this question. Is it true you can eliminate some of the arsenic from rice by rinsing it prior to cooking?
Yes. Wash it really well. You can get some out.
Excellent. Good to know. Thank you both. Again, this has just been a wonderful conversation. And we are now approaching the top of the hour. And so I want to thank you both for joining us and giving us so much to think about. I would want to say to both of you, there were far more questions on both your presentations than we were able to get to. So I hope you have a chance to glance at the chat afterward and perhaps offer some replies to some of the really fascinating questions that came through. You really stimulated a lot of thought from those who are joining us. I just want to note for everyone that these talks will be available on the MIT Tech Reunions website. That wraps up this first hour of brilliant conversation. We'd ask you now to stretch out, get a cup of coffee, take a moment, and then come right back here to this web address for the next hour's panel featuring professors Ed Boyden and Josh Tenenbaum. They'll be discussing new research on the human brain and how artificial intelligence can move a step closer to mimicking the human mind. That's just ahead in about 60 seconds. Stay tuned. And again, thank you for joining us.
Thanks for joining us. And for more information on how to connect with the MIT Alumni Association, please visit our website.